Everyone, it's really a great pleasure to present to you today about a revolutionary method to determine muscle and tendon function in freely moving animal and human systems. And before I move on, I want to talk a little bit about myself and give you a little bit of background. So I grew up in a small village in Belgium, in Koolskamp, as you can see here in the picture. It's very flat, there are no mountains, and it's very close to the ocean. And I always enjoyed science and mathematics because of the logic thinking behind it. And next to that, I also really enjoyed doing sports. And when I had to decide where I wanted to go to do my studies at the university, someone told me and gave me the recommendation that whatever you choose, just make sure that it's something you like, because you will have to do it for the rest of your life. And this was really a great recommendation that I got. And this made me to study the bachelor and master in physical education and movement sciences at the university in the beautiful city of Ghent in Belgium. And there I got introduced to the field of biomechanics, which is really the physics of the human body. And it's combining my interest in the human body, movement, and also in science. So this stimulated and motivated me to study an additional master's at the Liverpool John Moores University. And this master's was really research focused. So every class was almost a separate research project, which stimulated me as well, and which is really when I knew that I wanted to stay in research and continue specifically in the field also of injury prevention. From there, I went on to Switzerland, to the University of Lucerne and the Swiss Paraplegic Research, where I studied my PhD in health sciences and biomechanics. And this is also what brought me here at the University of Calgary, where I'm doing now my postdoc in kinesiology. But so my whole life, I had been interested in muscles, and muscle connects to tendons, to bones, and how the muscle is functioning will affect the tendon mechanics and also the other way around. And I'm really fascinated in how these two interact. And I wanted to get a more in-depth understanding of the musculoskeletal system. First, I want to give you a little bit, a short introductory video. Let me know if you cannot hear it. I hope you can.
Each time you take a step, 200 muscles work in unison to lift your foot, propel it forward, and set it down. It's just one of the many thousands of tasks performed by the muscular system. This network of over 650 muscles covers the body and is the reason we can blink, smile, run, jump, and stand upright. It's even responsible for the heart's dependable thump. First, what exactly is the muscular system? It's made up of three main muscle types: skeletal muscle, which attaches via tendons to our bones; cardiac muscle, which is only found in the heart; and smooth muscle, which lines the blood vessels in certain organs like the intestine and uterus. All three types are made up of muscle cells, also known as fibers. Bundles receive signals from the nervous system that contract the fibers, which in turn generates force and motion. This produces almost all the movements we make. Some of the only parts of the body whose motions aren't governed by the muscular system are sperm cells, the hair-like cilia in our airways, and certain white blood cells. Muscle contraction can be split into three main types. The first two, shortening muscle fibers and lengthening them, generate opposing forces. So the biceps will shorten while the triceps will lengthen or relax, pulling up the arm and making it bend at the elbow. This allows us to, say, pick up a book, or if the muscle relationship is reversed, put it down. This complementary partnership exists throughout the muscular system. The third type of contraction creates a stabilizing force. In these cases, the muscle fibers don't change in length, but instead keep the muscles rigid. This allows us to grip a mug of coffee or lean against a wall. It also maintains our posture by holding us upright.
And So we have the musculoskeletal system, and I'm really interested in how we can strengthen the system so we can prevent injury, and that we can have more people being active in a responsible way, and also how we can know when we're overloading and when we're at risk for injuries. And to study all of this, I went on to do my PhD in Switzerland, where I worked with wheelchair users. And these wheelchair users had a spinal cord injury. So a spinal cord injury is an injury to the spinal cord that can happen from a car accident, or if you fall from stairs or you have a skiing accident. This can also happen from a tumor, for example. And once you have an injury to the spinal cord, this causes significant loss of functioning below the level of injury. And this means that, for example, if you have the injury in this area where we have the green part here, so at the your core, for example, you may not be able to use your legs anymore, and you may need a wheelchair for mobility and activities of daily living. If you have then a higher lesion in your neck region, this can also affect your upper arms, and you may need additional assistive technology to help you with reading, opening doors, eating, etc. So it's clear that from the day of the injury to the next, these persons immediately become totally dependent on their upper extremities for activities of daily living. But the problem is that our upper extremities are not really made for these types of activities. Our shoulders are not designed to really handle these loads during this propulsion, when we have to transfer, moving the body from one place to another. And then the problem is evident that a lot of these wheelchair users have shoulder pain. And most of the times, this shoulder pain is related to problems with the shoulder tendons that run in this specific space between these two bones. So we have the upper arm bone here, and then this is part of the shoulder blade. And during these activities like propulsion, this space is reduced because of the position of the bones, and this irritates the tendons and may cause pathology and pain. And once these persons have pain, they cannot just rest like we would be able to do. They have to, or they will be immediately isolated from their family. They maybe not be able to go outside again because they cannot use their wheelchair for mobility. And this is why we really want to prevent injury and pain in this population. And if we want to do that, we have to have a closer look at these tendons. Tendons, they transfer these forces from muscles to bones, and they also store and release energy. Now, the good thing is that they have a high metabolic activity, and this means that they can adapt to the changing demands. Here we can see a tendon that is comprised of fascicle fiber all the way down to the collagen fibers. And a tendon, it's really like an elastic, as you can see here. And if we want to improve its injury prevention, we need to understand the failure properties and the capacity. Here we have the tendon. And when the muscle will contract, this will affect the tendon, of course, and go from totally slack to just not slack anymore. If we do that, we are within this range of the 0 to 1%. If the muscle is then contracting even more, the tendon will stretch. If we do this between 1 and 3%, this is normal. This is within the normal range. And when we do activities, it happens all the time. Our tendon stretches and relaxes. However, if we do, for example, activities like propulsion, which we do every day when we're in a wheelchair, and we transfer and continuously we have these loads, we can go over this range of this 3%. And then we're between the 3 and the 8%. And this causes injury to the fibers, the collagen fibers, and over time this can result in chronic adaptations. And when we really stretch too much above this 8%, we can even rupture the tendon, and then the tendon is detached from the muscle to the bone. And we see these chronic adaptations, we can see that with the use of an ultrasound device. So here we can see an ultrasound device where we're looking at the shoulder tendon of a wheelchair user. On this picture here, you see a biceps, and this is then the long head of the biceps tendon, where we're looking at with our ultrasound device. When we look at persons who have no pain, who are healthy, no wheelchair users, we see something like this. And this is the biceps tendon here in between these dark whites or clear whites horizontal lines. What you see is these nice and distinct black and white lines that represent a healthy tendon. If we then look at the wheelchair user's tendon, which is something like this here, we will see that the fibers are not so nice and clear. We see some dark, there may be some fluid inside the tendon. The tendon looks thicker as well. So there are these chronic adaptations that may play a role and that may cause pain. However, these adaptations may also be positive because we see these adaptations to also strengthen the tendon. But we don't know how we go from here to here. We don't know if they are bad. We know that these changes seem to be like there is degeneration that is not good and that may cause pain. But we don't know how we can really strengthen the tendon and make sure that we induce adaptations that may also make the tendon thicker and stronger, but then not weaken the tendon. This is why for my PhD, I then went on to look at these wheelchair users and to see what happens with their tendon if we let them do a very fatiguing, exhausting task. Here they are propelling along this H-shaped course for 3 times 4 minutes. As you can see, they make a turn, then they have to go as fast as possible to the middle where they make a complete stop, and then they have to accelerate again, and they're really pushing hard. We let them do this for 4 minutes, then they have 90 seconds of rest, after which they do again 4 minutes rest, and then they complete the last round of 4 minutes. And what we did then, we looked at how did their propulsion biomechanics change, did they change how they propelled, what's happening with their muscular activation, and also what is happening at the tendon level. What we found here was that following the fatiguing task, there was a significant overall reduction in the tendon thickness of the supraspinatus, or of one of the shoulder tendons. And this is a normal response to loading, which is alignment of these fibers in the direction of the applied stress with loading. However, we also saw that certain persons, those who are heavier, presented the opposite change. They presented an increase in thickness in their tendon, and this may be related to overload. These changes may then affect the capacity of the muscle and how the muscle is contracting, but this may also have resulted from muscle fatigue that was present after this fatiguing task. And it was really exciting, and we could do very interesting things by looking at these. We looked at 50 wheelchair users we asked them to do this protocol, and you can ask these persons, go to the maximum, how do you feel? You can do a lot of things, but if you do measurements with humans, you're very limited in what you can actually look at. So you cannot measure the actual force in the muscle, in the tendon. You cannot measure the stretch of the muscle or the tendon. You cannot see how the mechanics of the muscle and the tendon change at the same time. And if we really want to understand how these muscles and these tendons interact, and also what's causing this injury and this pain and how we can prevent it, we need to have a more fundamental understanding. And this is really what my goal is to do this postdoc in Calgary, is to get a more fundamental understanding of how these muscles and these tendons interact. We want to answer this question, and we will do this with something that has never been done before. And we will use a revolutionary method to actually measure this muscle and this tendon strain during these dynamic movements. And we will also add a lot of additional technology to see if we're actually measuring what we think we are measuring. So now I will lead you through all the different types of technology that we will be using, and I will explain to you what each technology does and how we get to the results. And some of these technologies are fairly easy and have been used or are currently being used in almost every research center in the world. But some of the technology have not been used before or really rare and can only be used or only been used in a small group of people in the world. So we will answer our question with looking at or with using a large animal sheep model. Here you can see some beautiful sheep in the mountains in Switzerland. This is actually where I biked along quite often. And so we're using these sheep because they are nice animals, they're easy to handle, but they are big enough for the technology that we want to use. And also their muscles and their tendons look quite similar to those of humans. So they're quite representative. Now I come to this passive electronic strain sensor. And this sensor, it's what you can see here, has been developed by one of the best universities in the world at the ETH in Zurich in Switzerland. And the sensor is really just actually this brown part. And it's connected to a wire, which you can see here. And this wire is embedded in silicone, but when we would insert the sensor in the sheep, this will not, the silicone will not be there anymore. And here you can see how we attached the sensor to the tendon of the sheep. So how does this sensor work? The brown part that I showed before is made of all small wires. And if we run a current through a wire, this will cause a magnetic field. And if we then change the sensor, stretch it, the wires will also be stretched, and this will change the magnetic field. And all we do really is measuring the change in this field. So we're measuring the change in the frequency. Here, this would be the wire, and then we have the current that goes through, and when we stretch it, we change the frequency, and then we have a wireless readout from which we can then transfer the signal with Bluetooth to our smartphone, and then we can immediately see what is the stretch on the tendon in the sheep while walking, but later on, possibly also in humans, when we let them run and walk, we would be able to see it without any wires. And this is one of the things that makes the sensor so revolutionary, is that it's wireless and that it will be able to measure changes in strain during dynamic movements, also during jumping, during very explosive movements. And this sensor has been tested before in the bladder of a sheep. Now, one of the applications for that, as you may think of the wheelchair user again, they are also most of the times not capable to void their bladder. And how the sensor has been tested is if you put the sensor on the bladder, once the bladder is full, the sensor will be stretched, and then this can give a signal to the muscle that stimulates the muscle, and that will then be able to void the bladder. So we could get a signal, the person would be, oh, my bladder is full, oh, I press the stimulation and then I can void, because this is really also a very big problem in persons who are having a spinal cord injury. And if we can then also use it during other movements, we can answer our questions about how these muscles and these tendons interact. Now, if we want to know if we're measuring with this sensor what we think we're measuring, we have to combine it with other types of technology that we are familiar with, and that we can then compare those results to the results from this strain sensor, so the stretch that we measure. And these other technologies include sonomicrometry. What is this? This includes, or these are crystals, which you can see here, that are connected to the purple wire. So we have one of the crystals here, and here is the other. And these crystals can talk to each other. If one of the crystals sends a sound, the sound will travel to the other crystal, which will catch it. And because we know the speeds of the sound waves, And because we know the time that it took to go from one crystal to the other, we can then calculate the distance between the two crystals. In this video, I want to show it to you again. We can see I was moving the two crystals closer and further from each other, how then this line changes and shows us the distance between the crystals. And with these crystals, we can also look at the muscle mechanics. We will be able to see the fascicle length. And if this is important information, because if the length is longer or shorter, this will affect the force producing capacity of the muscle. If the muscle shortens faster or slower, this all plays an important role in the force producing capacity. And with the sonomicrometry, we can look at these muscle mechanics. The next technology that we will be using is called a force transducer. Here you can see a force transducer. It's the one that we made now for the sheep, so it's a big one. And it's this E-shaped transducer. And here you can see the force transducer in a frog leg. What this really does is, if the tendon is stretched, this will deform the transducer, which will then send a signal to our readout, and from there we can then see the change in voltage. So this is also not a wireless sensor, the force transducer. And this has been tested in frogs, in cats, also in sheep, but is something that is only being used in a couple of places in the world. And in Calgary, they're really experts in using the force transducer. And we want to combine this together with the strain sensor, but with this one, we can directly measure the forces. However, we need to also, after we measured the change in the voltage signal when the tendon has contracted, we will then have to calibrate the force transducer afterwards. So we will have to dissect the muscle and the tendon and then look at what is this actually force now with a device, and then we can relate the actual force to the change in voltage. Finally, we will also use electromyography. You may be familiar with this. It's something that's fairly easy and that has been used before. Muscles, when they contract, they result in an electrical signal. So they cause an electrical signal. And with electrodes, we can pick up this signal and see the muscular activation. And the greater the activation, the greater the muscle force as well, most of the times. And for example, the activation will also change when the muscle gets fatigued and so forth. And we have electrodes that we can put on top of the muscle, but we also have fine wire EMG, electromyography, which is with wires that we go in the muscle, and then we can more accurately measure the electrical signal. And this signal will then look like this, something like this. And then when we analyze the data, we can nicely see when the muscle is contracting, we see an increase in the signal. And then when the muscle is not contracting anymore, it goes back to the steady, to the 0 point. When we then combine all these methods together, we will have something like this. I made a drawing here. We will have the EMG electrodes, the sonocrystals. We have the electronic passive strain sensor with the readout system, and then the force buckle. And only the electronic strain sensor that measures the stress on the tendon and the muscle will be wireless. And if we combine all these things together, we can really get very unique and interesting information about muscles and tendons. Here is a bit how our experiment is looking like. So we will have the surgery, and then of course we need a recovery phase for the sheep to recover from the surgery. We can see when the sheep is capable to walk as before again, and then we will start with the motion tests. We will just have a look when the sheep is walking, trotting, and what's happening with all this data. And we then want to repeat these measurements over time for 4 weeks, after which we will remove all the technology, because we want to see how do these results change over time. Is the sensor consistent or maybe not? And as you can imagine, such project that includes so many different technologies has a lot of challenges, and a lot of things can go wrong, and it may not work out as we expected. However, we know the sensor worked in the bladder, and the University of Calgary is familiar with and our experts and the other methods. So by combining all this expertise together, we can really get to very interesting insights. And even if the sensor is not working in the end, it will provide us new and fundamental information and allow also to adjust the sensor in Zurich, so they can adjust the sensor and then we can test again. And ultimately we want to test it in humans. And I really hope that we can also at some point test it in the shoulder of these wheelchair users, so we can really see what is happening at the shoulder and how can we strengthen these tendons, when do we overload it, and how do these muscles and these tendons interact. So with this, I want to acknowledge the funding, the Early Postdoc Mobility Fellowship from the Swiss National Science Foundation, who allowed me to come here, the Host Institute, the Human Performance Lab at the Faculty of Kinesiology, directed by Professor Dr. Walter Herzog, who is also my mentor. And I also am very grateful for the support from technicians, from all the doctoral and postdoctoral students. And of course, very fundamental here is also the development and the provision of the passive electronic strain sensor from the ETH in Zurich, who was the Lab for Movement Biomechanics, directed by Professor Dr. Bill Taylor, is very supportive, and it's really nice to have this collaboration across multiple countries that will allow to come to these unique data collections and insights. So I want to thank you for listening. Please feel free to ask me any questions. I'm happy to open the discussion with you.
Thank you so much, Doctor. So if anyone has any questions, you can either unmute your mic or you can put your questions in the chat.
It's a lot of information.
Was there anything maybe that you can think to clarify? Oh, we got a question. Okay. So are there any ways to make sure we don't put too much strain on our tendons?
Yeah, that's a really good question. So that's the problem, that we don't know at the moment what is the perfect amount. We need, when we don't have any loading, what happens when we go in space, this has detrimental effects on our muscles and our tendons. So we need a certain amount of loading. It's very important. However, we don't want to overload. Now, we don't really know what is too much. What we know that is important is that we see these changes with loading, and it's normal that we have these changes, but if there is not enough time to recover and we repeat it over time and over time and over time, it's really this repetitiveness that will cause the injury. So that's what happens with the wheelchair users who are continuously propelling their wheelchair. They're doing these repetitive activities. It's what happens with a tennis player who is always doing the same movement with also because it's overhand. Also, when you do overhand sports, you are reducing the space, which is also what happens during the propulsion. So the tendons are more comprised. Therefore, what's mostly important is that to have this balance between loading, and if you feel there has been too much, that you take enough time to recover, and then you can load the system again. So you can build it up. But it's important to listen to your body and feel, okay, is it too much or not? But we don't really know from a scientific point of view what is exactly the perfect amount of loading. And this is really what I'm interested in to find out.
I'm sure that also varies from person to person depending on the health of their tendons, or is it?
Yes. Yeah. Yeah. And so it depends on the capacity of the persons and the loading that the person has. So in the wheelchair users, it will also depend on if you have a higher lesion level, you will have less functioning below the lesion, so you will more rely on your shoulders. If you are, what's also found is that, for example, females have more problems, in specific in the wheelchair users with shoulder pain, that also body weight. When you're heavier, you have more loading. If you change your propulsion style, the way how you propel a wheelchair, you can propel with short strokes, so you push and then you go backwards. This is more demanding for your shoulder than if you do really nice and smooth long strokes. So they're really current guidelines include propulsion styles that lower the loads on the shoulder, also strengthening exercises. If you have stronger muscles, stronger tendons, you will be able to endure greater loads. And then it's also important to be aware that if you are if you're heavier weight, that you will have greater loads on your shoulder, so that you try to lower your body weight. And in that way, you can then improve or try to prevent injury from happening.
Okay, thank you. Does that answer your question? Oh, so tendons don't develop the same way as muscles do?
Yeah, indeed. So muscles and tendons are very different structures, and tendons really, if I want to go back to this pictures here. So in the tendon, we really see, you can see it as well. So it looks very different. So the tendon is comprised of the collagen fibers, and the muscle is, the active muscle will contract, and then the tendon will really transfer the forces that the muscles produce to the bones. So the tendon is actually resulting more for passive, for transferring this force over to the bones, while the muscle is really actively contracting. There's a lot of things happening in the muscle that you get the stimulation from the brain to the muscle, and then different things will happen. There's release of ions that will activate it, and then the muscle is contracting, producing a force, and then this will be transferred through the tendon to the bone. And in tendons, we will see these changes in these collagen fibers. And in muscles, we can see when we make them stronger, you can see it, someone who's really strong, the muscle gets really much bigger in size. And we will have less of these chronic degeneration in the muscle as we see in the tendon.
Okay, so there's another question. So does the tendon contract at all? Like, is there any give, or is it just like a rigid structure?
Yeah, so the tendon will change its formation, but it will not contract itself. It's contracting because of the changes in the muscle. If that's yeah.
Does anyone have any follow-up questions to that information? Or any further questions?
So one of the things that we also looked at is, I was also invited to conduct research now with wheelchair athletes, and it's really interesting to compare these persons who are highly active and athletes compared to persons who are also in a wheelchair but less active. And what we want to do, because these athletes will be stronger, but they also have to endure greater loads because they're always training and they're doing these repetitive forces. And what they want to do in Loughborough, which is where I went in the UK, is they want to add such ultrasound measurements to their yearly screening. For example, at the moment they are doing physiology measures, they're looking at their aerobic capacity, how fit are they, how strong are they. And now what they also want to include is these measurements of their shoulder tendons and see how they develop over time. And this will be something really interesting as well, because I for my PhD looked at very acute changes in these tendons. But if we then look over time and see, okay, these persons had these kind of training, this is exactly what they did, how is this now affecting their tendon?
So another question, are there certain tendons that are weaker than others?
That's a good question. So our body is developed in a way that it's efficient, but also not breakless, because it has to be efficient. So what is expected is that the tendons are as strong as they are supposed to be for this function that they have in the body. For example, here we have the Achilles tendon on this picture, which is known to be a very strong tendon that stores and releases a lot of energy. If we look at this, for example, the kangaroos who are really jumping, they have very compliant tendons because they store the energy and then they release it. And so yes, there's definitely a difference between all the tendons in the human body, but they all consist of these collagen fibers and they have the same function.
Does that answer questions? We still have some time left, so if you want to clarify.
I may have a question. Does any of you have injuries before and have any experience with shoulder problems or any other injuries to your tendons? No. Or any ideas on
Are tendons the same as ligaments?
So tendons are not the same as ligaments, because ligaments really provide passive stability to the joints, and ligaments are connecting bones to bones, while the tendon is connecting the muscle to the bone.
So if someone's had a strain in their Achilles tendon.
Okay. And how did, do you still have problems, or did you recover, or you want to share your experience?
You could even turn your mic off of mute if you'd like to talk about it. Or you can put it in the chat and I can read it, no problem. So they said, it hurts sometimes when I run long distances, but otherwise it healed.
Okay, that's really, really nice to hear. So, and have you changed your way of activity? Are you doing less, or?
So while they're typing, so does this type of injury usually heal completely, or are tendons less likely to heal?
So
like skeletal injuries or muscle injuries?
tendons will heal less. Well, they may not completely heal, but you may be able to remove the pain. And with that, it's because it's not because we see pathology that the persons necessarily have pain. And if you improve, if you do your rehabilitation training and you improve the structure, you can become pain-free, but you may not be able to have exactly the same tendon as you had before the injury. So it will not completely heal, but you can definitely work on it and become pain-free. The problem is that the treatment strategies are still not clear, and there's still a lot of unanswered questions that we have with the rehabilitation of injury. So we know that these exercises help and improve functioning again, but we don't really know exactly what we have to do. So we really need an answer to all these more fundamental questions.
Yeah. So the answer to your previous question, the person who had the Achilles tendon strain, they said they still run the same amount that they used to, but has changed up their routine, their warm-up routine.
Okay, so that's really, really good. And that's indeed, it's really important that it's not because you have an injury that we stop moving and stop exercising, because it's really important. We need these tendons and our muscles, our body needs this loading, and we need to find a more a better way to move. And if you can adjust your warm up, and this helps to warm up your muscles and your tendons, and you don't have pain anymore, that's really great. That's really wonderful.
Are there any further questions? I think we'll give it another couple of minutes to see if anyone can come up with anything. Okay, so do the tendons of most animals resemble human tendons?
Well, so the tendons in animals have the same function as they have as the tendons in humans. They differ in their size and in their, also because the anatomy of the animal, as you saw in the picture that I drew, the Achilles tendon of the human looks still different to what we see here in the sheep, but they have the same function. And so that's what actually allows us to really look at these, to do these measurements in the sheep, because they have the same structure. They also consist of the collagen fibers. They will also degenerate over time. They will also adapt to the loading. The difference is that the loading will be different. The way how they walk is different. There are a lot of differences, but it's still is the same type of structure. And this is really the nice thing that we can do when we do this animal research, is that we can do all these measurements that clearly we would not be able to do in humans. I don't think any of you would want to have such sonocrystals in your muscle, to have this, especially the force buckle is something that we cannot do in humans because we have to dissect the muscle afterwards. So yes, that's what really allows us to look at the same structure in the animals.
So a lot of thank yous, a lot of thank you. It was very interesting. So it's like our participants really enjoyed your talk.
That's nice to hear.
Yeah.
If you have any more questions, feel free to contact me. I can show you my email address again. I'd be happy to have a chat, or yeah, if you have any questions, let me know.
Okay, thank you so much, Doctor. I think we're going to end the talk here. Unless someone wants to squeeze in another question. I don't think there's going to be any more. But you can always email Dr. Bossuyt if you have any questions. Thank you so much again. Thanks everyone for attending. You guys were really interactive. Okay, have a good
Thank you for all the questions. It's really nice to talking to you.
Okay, bye-bye. Have a good evening, everybody.
Bye.